And we have our last speaker here. You need to give your microphone back, please. Thank you. Please, can I introduce Dr. Ina Shomer from the Sheba Medical Center of Israel. And I think this is a perfect talk to conclude this very interesting session in which we learned a lot about mechanisms, models, and so on, by hearing some more about therapeutic perspectives in this disease, and in particular in its working on RNA-based therapeutics, which is a field that is really exploding over the last very few years. So we look forward to your talk, Ina. Thank you.
Okay. So first of all, thank the organizers for inviting me to present our work. Oh, I need this. Okay. Where? Okay. So in Sheba, we have a 5-year-old female patient. She has severe hypotonia and dystonia and global developmental delay. She harbors the E246K mutation, but in addition to the known pathogenic variant, she has additional SNP. She has no seizures, normal EEG, normal MRI, and she's clinically monitored by Professor Brulia Benzev. In Sheba, we use the ASO approach for RNA-based therapy. ASOs are short RNA sequences which are complementary to the target RNA. They are chemically modified, which confers their stability, it reduces toxicity, and it affects their mode of action. And there are 2 modes of action, mainly 2. One, it activates RNase H-mediated degradation of the target RNA, and the other one is steric block. It can block splicing sites. And many pharma companies today are developing ASO drugs. The most famous one is Ionis, and they already have approved drugs and many drugs in their pipeline in clinical studies. In 2016, the first ASO drug was approved. It's called Spinraza for SMA. And it opened a new window and a new opportunity for development of more ASO drugs. 2 years later, the first N-of-1 ASO treatment was given. It was developed by Timothy Yu in Boston Children's Hospital. It was developed for a girl called Mila. This is why it's called Milacin. The compound was called Milacin. She had severe neurodevelopmental disease called Batten disease. And because her condition was very severe, and also by the presidency of the development of Spinraza, Timothy Yu developed a specified drug for her, and it was very quickly approved by the FDA and was given to Mila. And that encouraged additional research group to participate in the new form of personalized medicine. So today there are several organizations like the N1 Collaborative, 1M1M, and the DCRT. Okay. In Sheba, this is our pipeline. Okay, so first, after the diagnosis, we design a library of ASOs, these short sequences. We do in vitro screen. Next, we test the ASOs in patient cells. In some cases, we have a mice model. We do the tox studies, and next, we hope to treat our patient. Okay. So first, the ASO design. We were very encouraged by some publication that haploinsufficient patients have much milder phenotype, and we decided we can try to reduce the mutant allele by the ASO. We designed the ASO around the site of the mutation. The ultimate goal was to reduce the mutant allele and to keep the wild type intact. So the ASO was designed as a gapmers. You can see the scheme of the ASO here, and usually the mutations sit on the gap site. We screened 45 ASOs using the in vitro screen, in which we use the PsyCheck plasmid. In this system, we take 35 bases approximately around the mutation. We insert it adjacent to a luciferase reporter, separately the wild type form and the mutant one, and we monitor the luminescence. So we did this for our 45 ASOs, and we chose 10 best candidates for next screen in patient cells. Okay, now we took a biopsy from our patient. After that, we derived fibroblasts. We reprogrammed them to iPSCs, and we differentiated them to NPCs. These are this small ball of cells, and then to neurons, to DRG neurons. And as we all know, GNAO1 is mainly expressed in the neuronal cells. So we wanted to test our ASO in the most compatible environment. What we saw, first we wanted to characterize patient cells. So we saw that the mutant allele is expressed half the amount of the wild type one. It's not in the same level of expression. Also, we saw that in patient cells we saw reduced differentiation markers and increased in the sphere size, in the NPC sphere size, meaning the cells are more proliferative, less differentiated. We developed isogenic heterozygous knockout cell line from patient cells. We knocked the mutant allele because we want to be more sure it will be beneficial for the patient, this kind of treatment. And we saw increase in the differentiation markers and also reduction in the sphere size, in the proliferation. We also did RNA-seq and monitored differential expressed genes, and we saw that the isogenic wild-type null cells were more similar to the wild-type cells than to the patient mutant cells. So encouraged by that, we continued and tested the best 10 ASOs on patient cells. We differentiated them, and we monitored wild-type allele comparison to the mutant wild using real-time PCR. We did that using universal reverse primer and specific forward primer. We also confirmed the results using RNA-seq, targeted RNA-seq for GNAO1. And we saw a reduction in some of our ASOs. So next we continued and we chose 4 best ones and did dose response. And you can see, for example, ASO 41, a nice reduction in the mutant one, and the wild type was completely intact. Okay, so next we need to do the toxicity and the functional assays. So first we did the toxicity prediction assay. It's done in BJAB cells. This protocol is published by Ionis, and BJAB cells are a good model to predict toxicity, the immune toxicity. The red one is the toxic control, and you want your assays to be below that. So we had 3 candidates. And we chose to continue the functional assays using ASO 41, which was the lowest one. And we monitored cyclic AMP levels. You can see that patient cells show increased cyclic AMP levels. And when we deleted in isogenic wild-type null cells, you saw a reduction in the cyclic AMP levels, and also following treatment with ASO. Next, we used the RTCA system to monitor cell proliferation. We seed the cells on this special molds inside this the RTCA system machine, and it counts cell index. Okay, so the black one is the wild-type cells. The blue one is the mutant cells, the patient cells. The gray one is the isogenic control, so you see it reduce the proliferation. And the red one is our ASO-treated cells. Okay, so next we developed the mouse model. Luckily, the sequence around our mutation site is completely similar, and it's very promising because we can test our ASO directly on the mice cells and in mice brain, because it's sequence complementary. So we have now this mouse model for E246K. And at the beginning, these mice don't show much phenotype. There were no abnormal behavior, no abnormal features. But when they reached 3 months, they showed 30% of them showed neurological symptoms, spontaneous or in response to mild stress, like handling. So we need to handle them very carefully. They showed link clasping induced by tail suspension. They have dystonic posture. They have seizures. And approximately 20% of the mice died following the seizures, the following days. And they also have abnormal motor behavior. And sometimes they have this temporal inability to move. So we extracted neurons from mouse embryos, day 14, and we treated them with our ASO compounds in increasing dose response. And we saw promising result, especially with 41. And now we are in the stage we are injecting mice pups with ASOs, and we collect samples. We monitor RNA expression, and we also hope to see some beneficial results regarding phenotype. We also started tox studies in rats, so we started single-dose tox studies, and we also have promising results. And to summarize, we have a 5-year-old patient, female, with a de novo E246K mutation. And we were encouraged by sporadic publication describing a milder phenotype in patients. We hypothesized that allele-specific reduction of the mutant can reduce the phenotype. We showed that knockout of mutant allele in patient isogenic cells might have beneficial effect. So we decided to reduce the mutant allele using a specific ASO. Using a known chemistry, so it will hopefully be fast approved, we designed a library of 45 GAPMER ASOs and treated them using and tested them using reporter plasmid. We proceeded to test our 10 lead candidates using patient cells. We tested ASO toxicity and functionality. Next, we established GNAO1EU23 mouse model, harboring patient's mutation, which exhibited spontaneous stress-induced dystonic episodes and seizures. We tested our ASO on culture of NPCs from mouse embryos, and we did single-dose tolerability test in rats, which have a good response. And now we're in the process of regulation in the Ministry of Health in Israel, and also we're in the process of all the ASO manufacturing and the CMC protocols and all that. I want to thank our amazing team, and thank you. Oh, I need that.
Hey. Thank you so much.
Thank you.
This is promising and really gives new hope, to the families to hear that there are so many attempts to move towards treatment, which is the goal. Any questions? Yes, Alessandro.
Hi, great work. So you have shown that this ASO can let's say, cut specifically the mutant messenger RNA, right?
Yeah.
But have you checked if you have any effect on the translation of the wild-type protein? So what I mean, can it still bind the wild-type messenger RNA without cutting, but maybe interfering with translation?
Well, we didn't check that. You mean in the protein level? We didn't saw in Western, we didn't see any effect on the protein level. and it's hard to see the difference exactly between the wild type and the mutant on Western, right? Maybe we should check that, maybe mass spec.
Yes, please, Vladimir.
Thanks a lot for the presentation and the great work. I am puzzled about this additional SNP, which was present in the mutant allele. So the first question is whether you think this SNP may be responsible for the reduced levels of the mutant mRNA expression.
Like that it's half of the...
Yes.
It might have.
And the second question is this SNP also seen in other patients harboring the same missense mutation, or is it unique for this particular patient?
It's, as far as I know, it's unique for her. We don't see other patients with that.
Thank you.
So it's never been reported?
The double SNP? Like the no.
Okay.
Maybe it exists somewhere, like we don't know. But.
Okay. And can I ask you, what do you expect would be the administration of this ASO? Is it something that can be done, let's say, one shot, or it would be something that had to be repeated? What's your scheme?
Well, ASO usually are given, first it's in small dose, minimal dose, and it's incremented if everything is okay. And eventually, the patients are treated once every several months. It's given intrathecally, like.
So it's like a similar approach like for SMN, for instance?
Yeah.
Exactly. Okay. So any other questions? No? Okay, I think we can close this beautiful session. Thank you very much to all the speakers. Thank you to the organizers for putting together such an exciting program. And I will